Okay, welcome back to the final lecture for week 5. We're going to continue on with antimicrobial chemicals. So we looked at interferon at the end of the last lecture, and we're going to talk about complement with this one. So let's take a look at this. We're going to go past interferon, and we know that this one, again, it's an innate immunity. It's not specific. There's no memory. However, if a body cell is infected with a virus, they can actually produce this interferon alpha. It's going to go out to the cells, those healthy cells. It's going to cause them to produce antiviral proteins, and it's not going to allow those viruses to replicate in there. So it will help protect our healthy cells from that. And you can see, if you look at this one, it also attracts and stimulates natural killer cells to the site, so it'll get rid of those unhealthy cells as well, those viral filled cells. Okay, so we're moving on to complement. Complement is a cascading reaction, kind of like blood clotting was. So once one protein gets started, it causes another protein to become active, and so forth and so on. We're not going to look at a lot of the details of this. I have some basic things I want you to know about it. So it's this cascading reaction which causes the destruction of a target cell's membrane. It produces inflammation, and it can attract phagocytes and enhance phagocytosis. When we look at complement, the complement system, it's composed of over 30 different complement proteins. So we have C1 all the way up through C30, and we're really not going to spend a lot of time looking at these various parts of this one. There are two different ways that complement can function. The first is called the classical pathway. This is the one that we understood first. That's why it's called the classical pathway. It is also the most rapid and the most effective activation. In the classical pathway, you'll see here that here is my pathogen, and it already has the antibodies attached to it. So we're already involved in a specific immune response in that an antibody has been created against this specific pathogen and has attached to it. Complement, though, is an innate one, so it doesn't care what antibodies are attached. It just recognizes that our body has antibodies attached to something. So it doesn't matter what it is, it'll do the same thing. So when the antibodies have attached, that's going to be the kind of the start, and complement factor 1 is going to hook onto that, and that's what starts the cascading event. When the cascade ends, what I want you to know, you don't have to know all of these different parts, but what I want you to know is that it ends with complement factor C3, or complement protein C3, being converted into C3b. And what that does is that's going to continue this cycle and cause the other complement proteins to produce an MAC, a MAC, which is a membrane attack complex. And I'll talk about that in just a moment. Okay, so it starts with the antibodies being attached, and that gets the pathway started. A complement protein finds that, and then it very rapidly cascades through this event to eventually produce this this MAC. Okay. The alternative pathway is slower, and we don't have, this bacteria has not yet been recognized by any specific immune responses. So it doesn't have any antibodies attached to it. We don't have anything dealing with it yet. And so what happens is when there is a bacteria in, let's say, in your plasma, okay, there are complement proteins that are just found in your plasma, and when they are near a bacteria wall or capsule, it actually causes this complement pathway to begin. Okay, so the alternative pathway begins with an unrecognized bacteria or parasite or a viral filled cell, and the complement proteins come near it, and it stimulates this foreign object, stimulates the activation of this complement process. Again, this complement pathway ends with complement 3B being activated, which then starts the process to produce a MAC. Okay, and so we end up with this MAC, this membrane attack complex. It works very similar to perforin in that it actually punctures a hole, but this time rather than just having to go through a cell membrane, bacteria have cell walls, so it has to be a much bigger hole that it makes in there, and it causes the osmotic balance to be out of whack, and that causes the cell to lyse. Also, by having these complement factors attached to it, it will help with opsonization. If opsonization is, remember, opsonization basically makes a bacteria more attractive to a phagocyte, so it tells the phagocyte, like, here's a problem, come and attack me. So it does that. And these complement factors, if they've gone through this pathway, will initiate histamine release from the mast cells. So it's not actually tissue damage on this particular case, but it just simply is because we recognize that there is a problem and the complement has gone through this pathway. It will cause the mast cells to release histamine. Histamine will make the capillary beds in the area more leaky, and the complement will act as a chemotaxis. It will draw those neutrophils and macrophages to the site of this infection and actually kind of enhance your immune response. All right, the last innate defense mechanism that we can talk about is going to be fever. And then the very last thing that I want you to do is compare and contrast innate defenses and adaptive defenses. I've been talking about it throughout this lecture. I want to make sure that you understand the differences between these two. So we're looking at the fever. We're going to look at the role of pyrogens. We're going to talk about the benefits of fever. When you have a fever, it basically says that anytime your body temperature rises again above 37.2 degrees Celsius, that's about 99.1 degrees Fahrenheit, they call that a fever. And we have pyrogens that are circulating through our system all the time, but the more pyrogens that are out in our bloodstream will change our thermostat that our hypothalamus keeps for our body temperature. So if we have more pyrogens floating around in our plasma as it goes past the hypothalamus, it will tell the hypothalamus that you need to turn up the heat, and body temperature will rise. Pyrogens can be released from active macrophages. So again, we go to that site of an infection. We've got a major infection going on there. I really need to get my immune response up and running. The macrophage is going to release pyrogens, or if it releases any kind of the cytokines that it has, like your tumor necrosis factor, that will also act kind of like a pyrogen, and it will cause the body temperature to go up. Okay, there are benefits. Low fevers, so in that 100, 101 range, even 102, aren't really concerning. A lot of times doctors anymore won't tell you to do anything about it, because what happens is when I increase my body temperature, in order for me to increase my body temperature, I have to increase my metabolic rate, because remember, metabolism is what produces heat. So when I increase my body temperature, I increase metabolic rate. That means that cells move faster. That means that enzyme activity occurs more quickly, and it's going to give me that benefit of getting to the infection faster, dealing with the infection faster, and hopefully getting rid of it quicker. Also, that increase in temperature may inhibit some pathogens from being able to do their functions. They don't work so well at higher temperatures, and so that might inhibit them to a point. Now, if you have a high fever, you're getting into that 103, 104, 105, that's when we have some concern, because now your temperature is so high that you can do damage to some of those delicate neural tissue cells and those types of things, and we don't want to do that. So that's when you're going to take measures to try to decrease that. And also, if you have a low-grade fever that you have consistently for a week or more, a doctor is going to start taking a look and seeing what's going on with that as well, because they're going to wonder, what kind of infection do you have that you can't fight it off, that you're constantly running this increase in temperature, that your macrophages are constantly active, and they're going to start looking to see where that infection lies. Okay, that ends the PowerPoint, but I want to show you, if you go to the back page of your outline, this is where you can take a look at the, let me make my screen a little bigger right now. Whoops, that didn't make it bigger, that just moved it out. Okay, so let's make it so you can see what's going on here. If you go to Mastering AMP, and yours may look a little different than mine, but you want to go to the study area of My AMP, and you'll end up with a screen that looks like this. And oops, can't see it. Let me move it over. Sorry, guys. Here we go. Down here on the MP3 tutor sessions, if you click on that one, it gives you, underneath chapter 22, it gives you the differences between the innate and adaptive immunity. If you click on this one, it gives you two options. You can either download the MP3 so you can play it somewhere else, or you can listen to it right from here. Click the play arrow and listen to it. Or if you're one that prefers to read rather than listen, they have the whole transcript for you so you can read through it. This should help you answer the questions in this chart. I'm not going to give you the answers right away. I want you to dig a little bit. And if you have questions, feel free to post them. I put a discussion board link in your week folder, so you can post a discussion board question if you have one, and we can talk about it. But I want you to think about innate immunity versus adaptive immunity in terms of, how do you get it? Are you born with it, or do you have to develop it over time? What's your first line of defense? What comes in second? Does it have a memory or not? So these are the things that I want you to think out. Work on filling out this chart. I can guarantee you that something similar to this is going to be on that next lecture exam. Okay, the only thing you have left to do after you finish listening to this lecture is fill that chart in the back of your notes, and then do the take-home mini test that's in your folder. And make sure you bring a copy of that take-home mini test along with you when you come to the week 7 lecture. Hope you all having a great day. I'll talk to you later.